Good afternoon, everyone. It's my privilege to invite Dr. Rupert for this on behalf of Bioclues, Bioservices, and GCCI. It's our privilege to invite Dr. Rupert. He's going to present on contextual tissue cytometry with AI, immunophenotyping, and quantifying the tumor microenvironment in situ. So we'll be. I want to introduce him. Dr. Rupert is co-founder of Tissue Gnostic Group in Austria, Romania, and USA. He is Chief Executive Officer in Tissue Gnostics Austria and Romania, Vice President of Tissue Gnostics in USA, as well as Area Manager of Tissue Gnostics China. And before founding Tissue Gnostics, he was a research scientist at Competence Center for Biomolecular Therapeutics in Vienna, and joint venture between the University of Vienna and the Novartis Research Center. And he's a co-inventor of the TissueFax technology. Rupert has always been significantly involved in research and product development, from system design to clinical testing. And he has successfully headed several joint research projects and with academic partner institutions in the fields of advanced computer vision, cancer research, stem cell biology, and personalized medicine. And he's graduated from University of Vienna in cell biology. He has more than 20 years of experience in microscopy and image analysis. In addition, he has been trained in software development. So I invite Dr. Rupert to deliver his lecture.
Thank you very much, Dr. Sunil, for the kind introduction. Thank you, Dr. Brash, for the invitation. It's my great pleasure to present this afternoon about tissue cytometry and its applications, including applications that require artificial intelligence. I might want to add that since October last year, I hold the position of a visiting fellow at the Queensland University of Technology in Brisbane, Australia, and this is where I'm based at the moment. So before we go deeper into the technology and the applications, it might be a good idea to make a step back and try to get the overview, what we are actually looking at, what we are doing. And in that sense, I would like to point your attention to this survey among 1,500 scientists done, which was published in Nature in 2016. And following this survey, there was a global discussion, also referred to as the reproducibility crisis. Namely, the result of this survey among 1,500 scientists was not more and not less than the fact that 70% of the researchers reported to have problems in reproducing published data. And another 20% reported that they have even problems in reproducing their own data. So the question is, are our published data in science only reliable to 10%? Which sounds not very promising. So following this survey, a call was raised for actually 3 items, action items. First, a better mentorship. This is something universities have to take care of. Second, the proper use of statistics. This is something each individual laboratory or principal investigator has to take care of. And third, more robust experimental designs. This is something where companies like Tissue Gnostics can come in. There is, in fact, a number of well-established technologies that can be used in order to quantify cellular markers, both on the protein as well as on the mRNA or DNA level. One such technology that has been available for decades is flow cytometry. If you go to Public Medline and search for this term, flow cytometry or FACS, you will literally find hundreds of thousands of publications. Flow cytometry offers a phenotypic and functional analysis of cells by quantifying immunofluorescence-mediated antibody staining on the single cell level. One major limitation to flow cytometry is that it can only be applied to cells in suspension. That's why it's called flow cytometry. As a matter of fact, the vast majority of tissue in the human body is not liquid, as we are all aware of, but it's solid. So you cannot analyze solid tissue sections in a flow cytometer, at least not in a proper way. The alternative technology that can be used for tissue sections on glass slides is image cytometry. This requires a slide scanner, scanning and digitalization of the slides, and then you can do the same type of phenotypic and functional analysis, just that the individual cells are now not in a cell suspension, but in a tissue section. And what I will refer to in this presentation is tissue cytometry by the tissue fax. This is the instrument and the way image cytometry is offered by Tissue-Gnostics, by my company. So who is Tissue-Gnostics? As it was mentioned during the introduction, I'm actually a cell biologist. As part of my PhD thesis, I developed the prototype of tissue fax at the Medical University of Vienna. And in 2003, the company was founded. So in 3 weeks from now, we will actually celebrate our 18th anniversary. The headquarters is located in the city of Vienna in Austria, so in the center of Europe. We have international subsidiaries and offices in Romania, Los Angeles, Massachusetts. Tissue-Gnostics Asia Pacific is a registered company in Hong Kong and has offices in Beijing, Taipei, and Brisbane. What I would like to point your attention to is the fact that we have now a list of reference publication. Research groups from more than 60 countries on all continents have published almost 1,700 scientific publications using our technology. And the company is also certified for ISO 13485, which is the main quality norm for medical products. And this has been so since 2008. We are working under the scope, or call it the umbrella, of personalized medicine, which is also called or referred to as precision medicine. The overall idea of precision medicine is that we accurately determine the cause of a certain disease. It's mostly related to cancer, but not limited to cancer, and understand the molecular mechanism which leads to a certain transformation or mutation in a patient, rather than applying standard therapies, right? Nowadays, standard therapies are applied, and the doctors subsequently see that that did not work, and then the next standard therapy comes. The idea of precision medicine is to first determine the exact molecular mechanism which has led to a specific form of cancer in a specific individual patient, and then choose the therapy, the matching therapy based for the mechanism working in that patient. And to do so, we need instruments, software, and technologies that allow to elucidate and to quantify such molecular mechanisms. In other words, we need to quantify proteins, but also genetic markers like RNA or DNA on the single cell level in tissue section. And this is the scope of Tissue-Gnostics. And to reach this scope and always stay at the forefront of scientific research, of technology, we have been collaborating with a number of academic institutions across the world, but I would like to mention in particular 5 major multi-million euro EU-funded Marie Curie innovative training networks, which dealt with digital pathology that was led by a university, de Castilla-La Mancha in Ciudad Real in Spain. Then the CASA biomedicine network, which focused on the calcium sensing receptor as a potential target of therapeutic intervention. This molecule plays an important role in a number of diseases, not only cancer, but also breast cancer or diabetes, asthma, and Alzheimer's disease. Then there was Alcatraz, Break Free from Cancer, a translational oncology network headed by University of Cambridge in UK. INSEM, the European Network on Cell Migration Studies. And those 4 projects have already finished. Currently ongoing is Helical, the Marie Curie project on big data analysis by machine learning for clinical benefit, headed by Trinity College in Dublin. The common denominator for all those projects for tissue gnostics was the need to have image cytometry solutions at highest level. And this is what we have prepared and also developed in the course of these projects. Now, why is it so important to quantify? I mean, all of us can look through a microscope and make a statement, like something is bright or not bright, or there is much or not much. But this is not what this is all about. Please have a look here on this slide. You will see there are different regions in different gray values. And when we look at them, we see some are brighter, some are darker. Now, when we look on A and B, my question to you now is, what do you think? What's the difference in terms of gray values? When we say gray value 0 is black and gray value 255 is white. So what's the difference between A and B? The human eye will never be able to say the difference is 37, right? But I have prepared here 5 answers. No difference. I'm sorry, this is an automated moving. No difference, smaller equal 20, 50, 100. Please guess what you think. The next slide shows, in fact, they're identical. It's hard to believe, but we can see here it's in fact true. This is an optical illusion. It comes from the fact that this area is surrounded by brighter, while this area is surrounded by even darker areas. And therefore we have the impression they are different, but they are not. And to be able to quantify on the single cell level how much of a certain molecule is present, that is what is tissue cytometry all about, similar to flow cytometry. And just to give you an idea how the instruments that tissue gnostics has developed look like, there are different versions, like here StrataFax 2, which supports bright field scanning of 2 slides. Or the main product that we have is called TissueFax. This is a family of products which is also available for bright field only, fluorescence only, or the combination thereof, and it's called TissueFax Plus. As you can see, this is based on a high-end microscope of Carl Zeiss. So Tissue Gnostics is OEM partner of Carl Zeiss Germany. It supports any kind of objective lens from 2.5x to 100x, including oil immersion, so automated scanning of the slides. The immediate result of this scanning process is what is generally referred to as the virtual slide. We can see here an entire mouse leg. So please note that this is not a picture taken with a webcam. This is the result of a scanning procedure at high resolution. Many individual fields of view are acquired and then brought together. They are acquired with overlap, and neighboring fields of view are stitched. That means pixel-wise matched, so that this virtual slide is visible. Like with Google Earth, we can navigate through the sample and zoom in to see the cellular or even subcellular details, like here into the bone. It works for all kinds of tissues, like also here we have fine needle aspirates or biopsies of prostate, and also in fluorescence. What we see here is a tissue section stained with 4 markers of a prostate cancer. We see here the individual glands in the of the prostate section with the lumen. Here is actually a prostatic stone or some sand. Yeah. In red, we see a marker cytokeratin. This is labels the secretory epithelial cells. Here in green, the basal layer are the secretory. These are the basal epithelial cells, and in orange is a marker that is a kind of tumor marker which shows at an early stage malignant transformation. The absence of a basal cell layer and the presence of this marker on the secretory epithelial cells indicates on the functional level malignant transformation, and that this is actually a prostate cancer. Also morphologically, when we just look on H&E, we might not recognize this. Again, we can zoom in here, then we start seeing the individual nuclei in blue. This is stained by DAPI, a nuclear marker, and even further than the details of individual glands, where we can really see individual cells, and those cells can be analyzed. And this is what the focus of my presentation is about. Just to complete the overview of the product pipeline, we also have inverted systems referred to as TissueFax I like inverted, where you can also scan slides by putting them upside down, or well plates. This can be combined with culture chambers, so that also live cell imaging, time kinetic experiments, and even and functional experiments in cell cultures can be made. For high throughput scanning, we have a slide loader that can automatically scan up to 120 slides. And each configuration of our product, each fluorescence configuration of our product line can be equipped with a spinning disc confocal to provide whole slide confocal imaging. We see here an example of a mouse brain section. So this is a section through an entire mouse brain. This is an example we received from Janelia Farm Research Campus of the Howard Hughes Medical Institute in USA. The rectangles, the individual rectangles that you see here are the individual fields of view acquired using a 20x objective lens. In order to acquire the entire sample, 350 fields of view had to be scanned. Each field of view contains 4 color channels, DAPI plus 3 antibodies. In order to scan the entire tissue section in a single focal plane, it takes 8.5 minutes. If you make a set stack, that means the main focal plane plus 10 additional levels above and 10 below. So in total, 21 layers, each layer consisting of 350 fields of view, each field of view consisting of 4 channels. That gives in total 29,400 images, or 115 gigabytes of data. This is automatically done, including autofocus and storing, in 1.5 hours. So this is still very fast. If you try to do this manually by moving the scanning stage always to the next position and then acquire 29,400 images, you will probably sit a few weeks and not 1.5 hours. Again, like discussed before, we can use this virtual slide to navigate through our digital sample, through our image data set, like in Google Earth, and then zoom in order to see the cellular or even subcellular details. This is another example of a mouse brain section, which I received from Jacobs University in Bremen in Germany. When we look into and zoom into this region, we see it's called the third ventricle. You see here the level of the detail, the degree of detail that is visible. You can scan the entire brain if you want at this magnification. You will have a huge data set, but you can then zoom in and see the details, zoom out, and have the overview. And this is all done on the same data set. Now, I have mentioned that TissueFax is based on the hardware The core of the hardware is a high-end Zeiss microscope from Carl Zeiss, Germany. Difference between a Zeiss microscope with image analysis software and a tissue fax cytometer. There are two main differences. The first one is the workflow that we have designed and the degree of automation. The second one is the quality of the image analysis, in particular the segmentation. And this comes with this flow cytometry-like data handling. Now, what is segmentation? I would like to explain this to this audience. We see here two examples. They have nothing to do with cell identification, but a lot with segmentation. The computers are great in handling images, but not so great in understanding what an image shows. In order to do so, we have to apply algorithm which find groups of pixels that belong together, and the algorithms create so-called measure masks, which identify which part of an image is one object, which is another object. And this is the art, the very art of tissue gnostics, to automatically identify single cells in tissue context, even when the cells are very dense and touching each other, which is the usual situation in tissue. Here you see that up to 20 parameters are available per marker. There are intensity-related or shape-related parameters. The analysis options that are available are called HistoQuest, which is histological analysis, single-cell analysis in histological samples, TissueQuest, single-cell analysis in fluorescence, and our high-end solution for contextual tissue cytometry is called StrataQuest. Now, let's go into detail and give you an overview of the applications that are possible. Quantification of nuclear markers is a basic application that we always need. We see here a standard staining of a colon with in an immunohistochemistry peroxidase staining protocol. In blue, we have the nuclei. In brown, we have a well-known marker called Ki67, a proliferation marker. It tells us how many cells are currently proliferating in this tissue. This is, for example, important in tumor diagnostics, in oncology, and this is used as the so-called Proliferative Index. Now, normally people look at this and estimate and may say, 10% or 30% of cells are brown positive, so are proliferating. We have also done our own survey among roughly 200 scientists and asked them how many cells are proliferating. In other words, how many blue nuclei are also brown. And the answer that we received ranged from 1% to 40%. So, which is a pretty big difference, if we are honest, right? This is an example of a very good illustrative example of visual evaluation and the main problems associated with visual evaluation. We always get a range of estimations rather than a reproducible measurement. When we analyze this sample with our TissueFax platform, then we use the nuclear marker, so the hematoxylin, the blue stain, as a master channel. And the yellow contours here show you what the software has recognized as individual nucleus. And then we quantify for each nucleus how much brown and how much blue, or whatever color is present. In this case, it's brown and blue. How much of each color is associated with each cell. And this is shown in such a scattergram, or people in flow cytometry call it a dot plot, where on the x-axis we show the mean intensity of the blue staining, and on the y-axis the mean intensity of the brown staining, so Ki67. This line here is famous in flow cytometry. It's called the cutoff, and it determines the level of specificity in our staining. Only cells with a staining intensity greater than the cutoff, in this case 20, will be referred to as positive. So we see here our positive population and below the cutoff our negative cells. And we also see that these are 14.5% of all cells which are Ki67 positive. Obviously, this number or this percentage depends on where you set the cutoff. So theoretically, you can generate any number that you want, but the question is, how do you have to set the cutoff? Well, there is a scientifically well-established method, also used in flow cytometry, and this is called the isotype matched negative control. So you have to use a separate tissue section where you apply an isotype antibody and you set and acquire under same conditions like your test samples. And based on this isotype, you set your cutoff and then apply the same settings to your samples under investigation. Then you can compare the results. And this way, you have an observer independent and quantitative result on the single cell basis. And whether you do the analysis on Monday morning, on Wednesday noon, on Friday afternoon, in India, in Australia, in Austria, you will always get the same result rather than a wide range of estimation that depends on who does the analysis and maybe at what time of the day, in the morning, in the evening, if you're tired or if you're stressed or whatever. So observer-independent quantification replaces observer-biased estimation. Now, this was the first example of nuclear analysis. What happens if our molecule is not expressed in the nucleus, or not only in the nucleus, but also, or maybe only in the cytoplasm, which we see here. This is an example from skin cancer research. We see here two biopsies. The first one is a biopsy of normal skin, and the second biopsy was taken after UV irradiation, so after sunbathing. The marker under investigation is called COP1, but that's irrelevant. We're talking not about the biology, but about the methodology to quantify these cells. And what we see here is that this marker is constitutively expressed in the epidermis, but after UV irradiation, the amount of positive cells and also the amount per cell increases by this brighter staining and by more cells being positive. Normally, people would look at this and say, this is 1+ positive and this is 2+ positive. And then maybe your colleague comes and says, well, I would call this 3+ positive. And then you discuss, and in the end, you decide it's 2.5+ positive. Rather than discussing how many pluses you assign to a certain sample, tissue cytometry will give you, again, an observer-independent measurement. We see here the sample of normal skin and here after UV irradiation. And what we see at a glance is that the number of positive cells increases from 16.8 to 44.7. So on the left-hand side, we have normal. On the right-hand side, we have the biopsy after UV irradiation. But what we also see is that the positive population in the UV irradiated sample shows a shift towards higher values. So what we visually perceive as brighter in this image is reflected here in a quantitative manner. This cutoff, again, comes from the negative control that we see here. This shows the level of autofluorescence or unspecific binding of the antibody, if any, occurs. And the insert, which is known as quadrant statistic, gives you the statistic of the... So this is the cutoff for the parameter shown on our x-axis, which is the intensity of the DNA marker. And this is the cutoff for the parameter on our y-axis, which is COP1, the mean intensity of this antigen under investigation. These two parameters, these two cutoffs separate our diagram into 4 quadrants: the upper left, the upper right, the lower left, and the lower right. And this is reflected too here in this quadrant statistic. We see there are in total 759 cells in this upper left quadrant. out of 1,697 cells which are present in total in this region analyzed. And this represents 44.73% of the cells being positive for a marker. One of the features that our clients around the world appreciate most is the ability to track back and forth individual cells from virtual slide to data space. So we have here, for example, a renal allograft rejection. The cells in green are T cells, in red are monocytes. And if we click on the green cell, we see it's blinking here, green positive, red negative. If we click on the red cell, it's green negative, red positive. So red is monocyte. Following our motto, from image to analysis, we consider this a step forward and therefore call it the forward connection, from image to data space. It also works the other way around. From data space back to the image, we can, like here, define a gate, which is a selection of a subpopulation of cells, and then highlight the content of this gate here in a specific color. So you can visualize where in the tissue section a certain subpopulation of cells is located. That means you get the connection between the phenotype and histology. The system not only tells you how many cells are there and how many express a specific marker or a specific marker combination, so co-expressions can be determined, but also where the cells with a certain phenotype are located in the tissue section. Many, especially people working in histology or pathology, are very used to looking at samples in the typical immunohistochemistry format, that means in transmission microscopy. Therefore, we have introduced what we call the Pseudo Ihc View. We see here a fluorescent samples with 4 colors, and we can digitally convert a fluorescence image into a immunohistochemistry image. So it looks like it was a peroxidase staining. It's actually DAPI, but it looks like hematoxylin. Then we add our green marker, our red marker, and the fourth here in this bluish. Here, it's a cytokeratin. So it is in fact a fluorescence image, but it's shown like it was an immunohistochemistry, so a bright field microscopy or transmission sample. And in fact, there is a 5th marker called SP red, but there are only 2 cells here and here which are yellow. This is the original image. This is the Pseudo Ihc view. A very important application of tissue cytometry is the ability that we can determine the immune status in a specific organ or in a specific tissue. With flow cytometry, we can determine the immune status systemically in blood. With tissue cytometry, we can determine the immune status in a specific tissue environment, in situ. This is also referred to as the phenotypic characterization of tissue infiltrating leukocytes. We see here an example which I received from University of Western Australia in Perth. It's a colorectal cancer stained for 4 markers: DAPI, T helper cells, cytotoxic T cells in red, and FOXP3. You may know that CD4 and FOXP3 double positive cells are referred to as the regulatory T cells. So yellow and green double positive are Tregs, regulatory T cells. Like explained before, we use the nuclear signal, the DAPI, as a master channel. And the green contours here show you what the system has automatically recognized as individual cell. So it is not that there's somebody sat there and draw these circles. This is all done automatically by the computer. And based on this segmentation result, automated recognition of individual cells, we can now measure how many cells are CD4 positive. This we see here, and in red, the backward connection. So there are some cells which have a lot of yellow, so a intense staining of CD4, they will be somewhere up here, because this axis shows CD4, and the further up a dot is, the more of the molecule of CD4 is present. Others have only a little bit of CD4, so a little bit of yellow. They will be somewhere close to the cutoff, but still above the cutoff, means still be referred to as positive. And all the cells with green contours are below the cutoff, so they do not have a specific staining of CD4, of yellow, and are therefore referred to as CD4 negative. We can do the same thing for CD8. And finally, for CD4 and FOXP3 double positive cells, right? We again have these two cutoffs. This is the cutoff for the CD4 marker. This is the cutoff for our FOXP3. And this gives us four quadrants. Here, the FOXP3 single positive. Here, the CD4 single positive. Here, the double positive. And here, the double negative cells. And the system tells you exactly how many cells in percentage, in total count, or in density, cells per square millimeter of tissue analyzed, are in which quadrant, so have which phenotype. Now, we have seen that we can analyze molecular markers, proteins, on single cell levels, be it in the nucleus or in the cytoplasm. We can also analyze subcellular components or structures inside the cell, like, for example, fish fluorescence in cytohybridization or RNA-scope. I would like to show you here, however, an example which might be of interest in particular for India, which is intracellular parasites. Yeah? It's the example of leishmaniasis. This has been developed in collaboration with the Institute of Immunology in Regensburg in Germany. I'm not going to talk about the biology of leishmaniasis, but about the methodology to detect the parasites intracellular. So leishmanias are protozoan parasites which are transmitted by a mosquito and infect certain cells like macrophages where they multiply. Patients suffering from this disease encounter really severe structural tissue damage. Now, in Regensburg in Germany, the group of Professor Ritter is working on a cure for leishmaniasis. And they used our inverted tissue fax system in well plates, in cultures of macrophages, to quantify the effect of treatment using different agents. What we see here is one virtual well, not a virtual slide, because we are working in a micro well plate. So this is one well of a 20 well plate. The individual rectangles highlighted here are the individual fields of view that had to be acquired in order to cover a full well. When we zoom in, we see here the blue dots, which are the big ones, are the nuclei of the macrophages, and the smaller dots here are the nuclei of the intracellular parasites, the leishmanias. Yeah. Now, we first used the TAPI stain, the nuclear signal of the macrophages, to identify all the macrophages. And then, for each macrophage, we generate a so-called ring mask around the nucleus, which we see here. This is done for all the cells. And inside this ring mask, we look for the dots, for smaller dots, for the nuclei of the parasites. Now, this algorithm is a mathematical procedure. It does not obviously really understand what the parasite is. It works with groups of pixels, uses intensity or texture parameters. So we need to fine-tune the algorithm and set its sensitivity. In the lower panel here, we see the control population without Leishmania major. So all the yellow dots we see here are in fact false positive. The algorithm here is too sensitive. We use this culture, this control, in order to set the sensitivity of the algorithm so that nothing is detected, because there are no parasites in this control. And then the same settings are applied to our test samples, which contain the parasites. And what the algorithm finds now, these are really the parasites. In this publication, comparison with manual counting was done, showing a very good correlation. We have also developed a detailed algorithm which is able to separate here touching cells. And this is of particular relevance for tissue, because in tissue, neighboring cells always touch each other by definition, otherwise it would not be tissue. So, and if we have here, because a lack between these two cells, as we can see here, is a lack of contrast, and the primary segmentation algorithm did not succeed to segment here two objects, but identified one doublet, a double cell. With a special detail algorithm, we close such gaps, and in the end are able to segment two separate cells and then count inside each cell how many parasites are there. And because there are two stains for the parasites, one which labels all nuclei, and from the second stain which only labels dead nuclei, we can see that those which are labeled in green are the living parasites, and those which are red and green double positive are the dead parasites. So with this methodology, we can not only identify single cells and parasites, but also determine parasites, intracellular parasites per single cell, but also determine how many parasites are alive and how many are dead, which is crucial in order to assess the effect of treatment. So, nobody of us has missed that we are living in a pandemic world. So I would also like to tell you that with our tissue cytometry platform, we have already supported virology research in many ways, not only since Corona, but already before. Like we see here an example of influenza and pneumonia, which could be a one-to-one copy-paste story for COVID, but this is influenza. And you see here that the viral load of parenchymal cell, of epithelial cells in lung parenchyma was quantified. So in red, we have the epithelial cells, and green is virus. So we can determine the viral load, how many cells, how many epithelial cells are actually infected, and how much of the virus is there. And in another application, or in another publication rather, Zika virus was analyzed. And here, individual virus particles were quantified per single cell. Or dengue virus. And with this application, I would like to emphasize what we see here, that with this tissue cytometry approach, we can not only quantify markers which are expressed on 50% of all cells. No, we can really work and look into minor cell populations, right? You see here 0.06, 1.39, 0.55% of all cells are positive for this dengue virus. So it's a very detailed and in-depth analysis, which is on the single cell level, in situ, and quantitative. I mentioned at the beginning that tissue gnostics, and not only tissue gnostics, but is working under the scope or in this or umbrella of precision medicine. Precision medicine is probably one of the current mainstreams for medicine into the 21st century. And as I mentioned at the beginning, precision medicine requires that we understand molecular mechanisms. Now, in order to understand molecular mechanisms, it's not sufficient to look at one marker, because cells, phenotypes, functions of cells are usually determined by a set of markers. And the more research we do, the more markers we need. So we need to look for strategies how we can increase the number of markers that we can analyze per single cell. And one of the strategies that has been developed, not by tissue gnostics, but is also supported by our instrument platform, is multiplexing. In multiplexing, we take a given tissue section, stain it with a certain combination of antibodies and fluorochromes, image it, scan it, and then remove the staining and restain the same tissue section with the same combination of fluorochromes, but in each staining round, the fluorochromes represent different markers. And subsequently, these images are aligned and registered. Registration is the term for aligning images into one stack so that we can analyze them at the same time. In this publication from Yale University, this process has been described using our TissueFax platform and StrataQuest software for this registration and cross-correlation, and then obtaining a very detailed analysis on the single cell level. We see here that like CD4, Ki67, granzyme B, interleukin 6, FOXP3, and other markers have been quantified, but the cells that you see here in all the figures from A to L, this is always the same cell population on the same tissue section, right? With some 10 markers stained, not simultaneously, but in a cyclic way using this multiplexing approach. Another technical approach to increasing the number of biomarkers or molecules that can be imaged and quantified simultaneously on the same tissue section, on the same cell population, is multi-spectral imaging. So a minute ago, we talked about multiplexing, which requires a standard fluorescence microscope and a cyclic staining and subsequent alignment of the individual images. In multispectral imaging, all markers up to 10 are applied at the same time and acquired by specific acquisition technology, which includes a liquid crystal tunable filter that allows to detect a lambda stack. When we talked at the beginning of this presentation about a confocal microscope, we talked about the set stack, which is, and where in a set stack, you look on a given sample, a given region, in different focus position. In a lambda stack, you look at the given sample in the same focus position, but in different spectral intervals. And by using a mathematical procedure referred to as spectral unmixing, the computer can calculate how much of the total spectral emission comes from fluorochrome 1, 2, 3, 4, 5, 6, 7, up to 10. And then we get here a very nice and clear and brilliant picture. And one very currently relevant application is shown in this Cell paper from Harvard University in Boston, where the immune response against the SARS-CoV-2 virus was quantified in situ in COVID-19 patients, so in the lung tissue of COVID-19 patients. And there was a 7-color immunostaining. I don't have the time to go into the details, but anyhow, it's a published paper, so you can always refer to it. And the unmixing was done using bispectral imaging, using our TissueFax Spectra platform. And just to make the long story short, this research had a rather unfortunate outcome for the whole world. Unfortunate for the whole world, it actually showed, there was the first indication last year that the concept of herd immunity against corona will not work, because the SARS-CoV-2 virus tricks our immune system and avoids the formation of germinal centers. In other words, prevents the formation of long-term immunity. And this is also what the authors of this paper is the main conclusion. Yeah? Now, we are well into the 21st century, into the age of computers. And of course, computer-assisted diagnostics, for example, has also played an ever more important role in our research and in our medical diagnostics. And in fact, tissue cytometry also offers solutions for machine learning or deep learning. For example, in our latest generation, which is called TissueFox Suite 7, we have a new segmentation algorithm which is based on deep learning. And this is especially important when you work with lymphatic tissue, where you often have extremely dense cellular conditions like we see here. This is DAPI only, and you can see that the nuclei are actually almost touching or effectively touching each other or overlaying each other. And this is a very, very challenging situation for the computer, sometimes even for a trained human observer, to see what is one cell and what is a neighboring cell. With this deep learning algorithm, we can obtain a really impressive result in the automated acquisition. Machine learning or classification is often used to identify certain histological structures, like here in the skin section. 7 classes are defined, including the epidermis, the dermis, or the subcutaneous fat. And based on this classification, further cellular analysis, as I have shown before, can be obtained. Now, we have seen that we can analyze or identify individual cells, the nucleus, the cytoplasm, subcellular compartments, or multicellular structures, all in an automated manner. Now we can combine this in order to establish a contextual image cytometry. Again, we see here a colorectal cancer. We identify all the nuclei. In orange, we see the measure mask for the tumor region. And then we can combine these measure masks and ask all kinds of questions. In this case, this research group from Perth has asked the question, how many CD4 positive cells, so T helper cells, are present? Where are they? Are they inside the tumor or in the tumor stroma? If they're in the tumor stroma, how far are they away from the next tumor area? And how many of the CD4 positive cells are also FOXP3 double positive, so are regulatory T cells? We see here a scattergram in which CD4 was used as an input gate. On the x-axis, we have the size of the nuclei, and on the y-axis, the distance in micrometer from the next tumor area. So each dot that you see here represents a CD4 positive cell. And the gate, or the position on the y-axis, indicates the distance in micrometer from the next tumor region. So we can make here gates which contain all the cells which are 1 to 25, 25 to 50, 50 to 100, and so on, micrometers away. The quadrant statistic shows us that we have in total 7,234 cells, and 2,267 thereof, or 31.3%, are in the first gate, which indicates a distance of 1 to 25 micrometer. Now we can use this backward connection feature that I've introduced earlier to visualize. Here, in orange, we see the tumor area, and in red, we see those and only those cells which are CD4 positive, are immediately outside the tumor and not further away than 25 micrometers. Or here, 25 to 50, 50 to 100, and so on. So we get a very detailed analysis in spatial context. And we can use this in order to analyze what is called the tumor microenvironment, in situ, on the single cell level, in a quantitative manner. So these distance zones can also be shown directly in the tissue in form of color regions, color areas overlaid. And then from those data, you can generate such graphs, like which shows the cytotoxic T cells, the effector T cells, and the regulatory T cells, and how many are inside the tumor or outside the tumor in the tumor stroma in a certain distance. So you get a very detailed analysis on the single cell level. Here again, we have 7-color immunostaining. It's one of those immune checkpoint marker combinations acquired with spectral imaging. This is the original image. After spectral unmixing, we get a very clear and brilliant image of this tumor. Here you see the individual regions, sorry, the individual channels. Here, this would be the overlay of all channels. Click on any of the cells, you will see it blinking in all the scattergrams. So you know exactly where that cell is located in your data space. And this would be the data set for the backward connection for each of the cell populations. I don't have the time to go here into too much details, but I would like to finally, as a last example, explain this to you. In the first scattergram here, we see the size of the cells and the PDL1 mean intensity. And here in this gate in green, we see the PDL1 positive cells. We now choose this gate as an input gate for our second scattergram. All the cells visible in the second scattergram only come from this gate. In other words, all the cells here are PDL1 positive. The markers shown on the second scattergram are CD4 and PD1. And we have here our 4 scattergrams as discussed earlier. If we now look on the double negative quadrant here, so CD4 negative, PD1 negative, and use this as an input gate on our third scattergram, that means we see here only those cells which are CD4 negative, PD1 negative, and PDL1 positive, because this was the input gate there in the second scattergram, and the lower left quadrant of the second scattergram is the input gate in our third scattergram. As the lower left quadrant is the input gate in our third scattergram. And when we now look again on the double negative here, which is CD8 and on the x-axis and CD68 on the y-axis, and make the backward connection, then we see here in red those cells which are PDL1 positive, CD4 negative, PD1 negative, CD8 negative, and CD68 negative. So we have 1, 2, 3, 4, 5 markers analyzed, co-expression of 5 markers analyzed on the single cell level in situ, in a tissue section. To conclude my presentation, I would like to mention that this system has been analyzed in many ways, also the quality of the data. The first one was a publication done by myself and our group back in 2006, already published in Cytometry. Here, the tissue fax is the name of the instrument. The name of the technology is microscope-based multicolor tissue cytometry, MMTC. And we compared here MMTC versus standard fax using peripheral blood leukocytes stained for a standard CD3, CD4, CD8 marker combination, and then compared the results showing a very good correlation coefficient on peripheral blood leukocytes between standard flow cytometry and tissue cytometry. The group in Perth, from which I have shown already data earlier, has also done such a comparison by manually counting FOXP3 positive cells in tissue sections of colorectal cancer. 3 observer manually counted defined regions. As you can see, and as was to be expected, each observer had a different output. That's very normal. This is what we showed earlier, the problem with manual evaluation. The median of the 3 observers was used to compare with the software obtained results, and this gives also a very high correlation coefficient between manual counting, which you can refer to as the gold standard, and automated analysis. Just that the difference is that the manual counting was done on a few small regions, while the automated analysis can be done on full samples with millions of cells in less time than the manual counting can be done in small regions. So I hope that with this presentation, I could give you an overview and show that you can use tissue cytometry to move from image to data and increase the observer independence and reproducibility. I would like to thank Well, I have already mentioned our reference publications. I invite you to go to our website, tissuegnostics.com. Under research, you will find a searchable database where you can look on which marker, on which disease, on which tissue type anybody somewhere in the world has already worked using this technology. And finally, I would like to acknowledge and thank our partners from academia, from our EU projects, but also our consultant from Brazil and our partner here, Professor Jyotsna Batra from Queensland University of Technology, with whom I am closely collaborating. And finally, I would like to thank you. I hope I could show you that with tissue cytometry, you can explore the fundamental unit of life, cells, at the precision that inspires. Thank you very much for your attention.